This talk is about ectodermal placodes. This talk will be conducted using a question and answer format. The first question is: ectodermal placodes are present along the whole neuroaxis. A true, B false. This is a representation of the ectodermal surface of an embryo of 21 days gestational age. In it, you can see delineated the neural plate. I have now introduced a thin bar labeled AA. It indicates the site of a cut at the level of the mid neural plate. In this new frame, you can see the corresponding image brought about by such a cut. In addition to the neural plate, the gray shaded area, I have added two dotted areas by the side of the neural plate. They represent the neural crest. You can now see the same surface at 22 days of gestational age. Notice the corresponding change brought about by the passage of time at the same level. This change consists of the appearance of the neural grooves. I have now introduced the transverse view of the area. In this view, the neural grooves can be better appreciated. In this figure, you can also see the neural crest and the somites. By 23 days, the grooves begin to fuse at the mid-region of the neural plate, forming the neural tube. In this new added figure, you can see the same structures as in the above figure, but with a different arrangement. Notice that the neural tube, the neural crest, and the ectoderm have become isolated structures. I have now introduced the representation of an embryo of 24 days. A cut at the same level shows the neural crest by the side of the spinal cord. The spinal cord at this level is formed by the neural tube. Now I'd like you to look at the top figure, specifically at the level of the rostral end of the neural plate, in which I have now introduced a thin bar representing the site of a new cut, which I am labeling BB. I will represent this cut at the side of the panel, as you can see here. In this figure, you can see the same elements, but in addition, I have introduced in light blue an area by each side of the neural crest. These elements correspond to the ectodermal placodes. Now let's go back to the representation of the 22-day embryo and again indicate to the rostral region and represent a rostral cut. The figure just introduced represents the elements present at this level. As you can see, they are the same elements as indicated in the figure above. We will do the same in the 23-day-old embryo representation, as indicated here and illustrated here. As you can see, the basic arrangement is maintained, and in the 24-day embryo, a rostral cut will show the brain, the neural crest, represented by the dotted lines, is apart from the brain and the ectoderm, and the blue element that remains in the ectoderm. I will further illustrate the difference between a mid-cut represented as AA and a rostral cut represented by BB by using more colorful illustrations. Here you see a mid-neural plate transverse cut at about 22 days. The orange bands represent the neural crest. Between them is the neural plate, and lateral to them is the ectoderm. The cylinder represents the notochord. Here by 23 days and here by 24 days. Notice the changes undergone by the orange area representing the neural crest. At the rostral level, in a 22-day-old embryo, you can see lateral to the neural crest in orange, the ectodermal placodes in blue. The new figure introduced here corresponding to a 23-day-old embryo, and here at 24 days, notice that unlike the representation of the neural crest that has detached from the ectoderm and the neural tube, the ectodermal placodes remain embedded in the ectoderm. So the answer to this question is B. Next question. Which of the following do not derive from the border zone? A. CNS. B. Neural placodes. C. Neural crest. From the embryonic ectoderm develops the non-neural ectoderm and the neural ectoderm. The non-neural ectoderm becomes the epidermis. The neural ectoderm becomes the neural plate. This neural plate forms all the CNS structures except the most distal portion of the spinal cord. From the border zone between the non-neural and neural ectoderm arises the ectodermal placodes and the neural crest. So the answer to this question is A. Next question. Which of the following hormones derives from ectodermal placodes? A. Gonadotropin releasing hormone. B. Antidiuretic hormone. C. Growth hormone. D. Corticotropin releasing hormone. E. Thyrotropin releasing hormone. This is the same figure we just built in the process of answering the prior question. The ectodermal placodes consist of a few isolated islands of special pluripotential tissue embedded in the rostral ectoderm. In this frame, I have introduced the different placodes. The adenohypophyse placode, the olfactory, neuron and gonadotropin releasing hormone placode, lens placode, trigeminal placode, lateral line placode, epibranchial placode, and the otic placode. So the answer to this question is A. Next question. Which of the following elements derives from the neural crest? A. Middle ear bones. B. Schwann cells. C. Autonomic motor ganglionic cells. D. Satellite cells. E. Melanocytes. F. All of the above. In this representation, we have the embryonic ectoderm producing 5 distinct masses, identified by their color. The light green corresponding to the non-neural ectoderm, the yellow masses corresponding to the neural plate border zone, and the cream color mass corresponding to the neural plate. The neural plate goes on to form most of the CNS. The non-neural ectoderm forms the epidermis. The border zone produces the placodes and the neural crest. From the neural crest, many structures develop. Bony structures arising from the neural crest are the frontal and nasal bones, zygomatic, maxillary, and dentary bones, escamosal, alveus sphenoid, malleus, and incus, iod bone and stapedius, and the thyroid cartilage. Peripheral nerve system elements, at least some, also arise from neural crest cells. Here I have introduced 3 histological specimens of different peripheral nervous system structures to point out some of the components arising from the neural crest. This is a specimen corresponding to a longitudinal cut of a nerve. In it, I am indicating a Schwann cell nucleus with its surrounding pericardium, and here to another one. I will now substitute this image of a longitudinal cut of a nerve by a transverse cut of a nerve, which I will enlarge, as you can see in this frame, to show you the Schwann cell forming myelin around an axon, and here a Schwann cell engulfing several unmyelinated axons. This is called Rimac bundle. These 2 examples can more clearly be seen using electron microscopy. In figure A, you can see a Schwann cell with its prominent dark nucleus and grayish cytoplasm myelinating an axon. In figure B, you can see a Schwann cell cytoplasm engulfing multiple axons. The axons are forming a relatively single row flanking one side of the Schwann cell's nucleus. These axons are unmyelinated. The structure formed by the Schwann cell and the unmyelinated axons is called, as we previously mentioned, Rimac bundle. I have brought back the transverse nerve cut, here indicated by the arrow, to substitute it with a higher power specimen of a similar cut, where you can see the dark pink area corresponding to the nucleus of a Schwann cell, cytoplasm, and myelin. The myelin is surrounding an axon. The specimen below corresponds to a sensory ganglion. Satellite cells, as indicated here and here, are of neural crest origin. I am now indicating to an autonomic ganglion. Satellite cells in the autonomic ganglion, indicated here and here, as well as the ganglionic autonomic neurons, are of neural crest origin. Notice that the nuclei are in the periphery, unlike the nucleus in the neurons of the sensory ganglion. So the answer to this question is F. Next question. Which of the following is true about cranial nerve 0? A. It is also called cranial nerve N. B. It is also called nervus terminalis. C. It is related to the vomeronasal organ. D. May relate to reproductive behavior. E. All of the above. This is the same scheme we introduced to answer the prior question, but this time we will use it to introduce the elements that originate from the ectodermal placodes. In this frame, I have introduced and label a representation of the ectodermal placodes. You do not need to read them at this point. We will discuss them one by one in the next few frames. We will start with the olfactory placode. Here you can see represented in yellow the olfactory mucosa, and in pink the nasal mucosa. A histological specimen obtained from the border zone is shown here. In this area you can see the olfactory mucosa, and here the nasal mucosa, with the impressive globed cells that histologically more than anything else distinguishes the nasal mucosa from the olfactory mucosa. I have reintroduced here the figure of the nose to call your attention to the olfactory bulb and surrounding area, which you can see further illustrated here. In this figure you can see represented the olfactory mucosa under the cribriform plate, the olfactory bulb, and the hypothalamus. I will use this figure to illustrate the origin of the gonadotropin releasing hormone neurons. In the olfactory placode, a subset of cells are gonadotropin releasing hormones precursors. These cells use the olfactory neuron axons to migrate through the cribriform plate and reach the bulb. Ultimately, they become gonadotropin releasing hormone neurons in the hypothalamus. Recall that gonadotropin releasing hormones are produced in the hypothalamus, secreted into the medial eminence, and transported to the anterior pituitary by the hypophyseal portal system. There, they induce other neurons to secrete FSH and LH. Here I have again and for the last time reintroduced the nose figure. I have done so to indicate to this element the controversial vomeronasal organ. This organ is innervated by cranial nerve 0, also called nervus terminalis or cranial nerve N. N stands for the first letter of the word nulla. This nerve is related to reproductive behavior. Obviously, the reason I mention this nerve here is because some authors believe this nerve is of olfactory placode origin. Other authors believe this nerve is of neural crest origin. So the answer to this question is all of the above. Next question. Follicle stimulating hormone and luteinizing hormones are secreted by gonadotropes, that is, gonadotrope secreting pituicytes. A true, B false. After the olfactory placode, I will briefly mention the lens placode. The lens placode, as suggested by its name, forms the lens. Next, we will talk about the adenohypophyseal placode. The adenohypophyseal placode gives rise to the Ranke's pouch, which form the adenohypophysis. The function of the adenohypophysis is dominated by a mesenchymal structure, the hypophyseal portal system. In the adenohypophysis, we find a conglomeration of rather specific secretory neurons. Collectively, these neurons are called pituicytes. All of them are of ectodermal placode origin. Pituicytes include neurons that release melanocyte stimulating hormone called melanotropies, thyroid stimulating hormones called thyrotropies, adrenocorticotropin hormone referred to as corticotropies, follicle stimulating hormones, and luteinizing hormones. These two hormones are produced by cells denominated gonadotropies. Neurons that secrete prolactin are called lactotropies, and neurons that release growth hormone are referred to as somatotropies. So the answer to this question is A. Next question. Which of the following derives from neural placodes? A. Middle ear bones. B. Schwann cells. C. Autonomic motor ganglia cells. D. Satellite cells. C. Cranial nerve ganglionic sensory cells. After talking about adenohypophyseal placode, we will briefly mention the trigeminal placode. The trigeminal placode forms some of the neural elements of the trigeminal ganglion. These are the pseudo-unipolar neurons, a fact that cannot be appreciated in this specimen. They are round in this specimen and have a central nucleus. This is another specimen in which you can actually appreciate the unipolar nature of these neurons. The arrow points to a single structure dividing into two neurites. Next, we will talk about epibranchial placodes. The epibranchial placodes give rise to the neurons at the geniculate, petrosal, and nodosal ganglion. The otic ectodermal placode and lateral line placode, represented here, form the inner ear. I will in this talk only emphasize some elements of the cochlea. This is a cartoonish representation of the cochlea. The structure indicated here and here corresponds to the spiral ganglia. These are the site of the bipolar sensory neurons for hearing. Notice that the body of this neuron and its two neurites coming out of it. These neurons are also considered of ectodermal placode origin. Here you can see a lower power image of a spiral ganglion. With good imagination, you can see the bipolar morphology of the neurons. Whether Schwann cells and satellite cells in this ganglia are of ectodermal placode or neural crest origin is uncertain. These comments made for the spiral ganglia present in the modulus of the cochlea can also be made for the elements inside the Scarpa's ganglia. So the answer to this question is E. Thank you very much for your attention.